All right. So, can you give me a brief history or any sort of highlights in your career that you think we should start off with?
So I trained in some very good labs. I had the privilege of training with very good people. First as a graduate student at the University of Chicago, and then as a postdoc at Stanford. And I would say that probably the strongest feature of both professors was their integrity. Their honesty about the data and how you analyze the data and how you present the data, and their honesty about applying for funding, that you don't make things up. So I would say honesty and integrity was a big part of the training.
All right. Yeah. And do you think that's something that's still around nowadays, or is it sort of watered down just a little bit, or do people?
I think it's watered down.
Okay.
And I think it's watered down for two reasons. You know, science moves very quickly now.
Okay.
And there's more pressure, I think.
More pressure.
When I trained, and in the labs I trained in, which might have been even a little bit unrealistic at the time, the idea was to take on a long-term project, find your own question, take on a long-term project in an area we didn't necessarily have competitors, where you could explore and examine and grow and take your time to learn and find unique things. And I think now there's much more competition.
Yes.
Many people are doing the same thing because they're using the same tools very often. So there's a lot of pressure on students to produce very quickly.
Very quickly. So is that a symptom of the fact that the low-hanging fruit has been sort of picked in most of these disciplines?
I think in a lot of cases that is a symptom of the low-hanging fruit. But also there's a selection process. I mean, a lot of the people who end up being young faculty now were probably picked because they could, publish very quickly. It's a mark of success.
It's a marker of
and
success.
And then, so their students, they push their students to keep up the reputation. There was one other point I wanted to make, and I forgot. What was the question again? What has changed?
What has changed in regards to research and how leaders actually sort of galvanize their students to...
And I would say teamwork, as science has gotten bigger, teamwork is more important, and that can be good and bad, but you end up very often in your training, you end up being a small person in a large lab that that lab needs to accomplish a lot. Nowadays, papers require many different techniques. There might be 2-photon, there might be transcriptome, there might be biochemistry, some programming, psychophysics, behavior. And so projects tend to be much more complex. It gets back to your idea of low-hanging fruit. And
so
you're
it's
in bigger
more
labs.
Applied now.
Yeah, you're in bigger labs, and the idea of individualized training, I think, is much more rare, where you have an advisor who actually spends time with you and tells you how they think.
All right, so I've got a question in regards to that. So in a big lab, how would you, as a leader in the lab, sort of make sure everyone is not left out? That's my first question. And my second question would be, even though you're maybe at the lower rung in this big lab, how could you still, exhibit leadership qualities, even in a lab where you're maybe an undergrad, or
how could the student exhibit?
How could the student? So two questions there. How could
leadership
the
qualities?
PI?
So, organizing groups is a big challenge. Organizing groups in research is a big challenge. You're not making widgets. It's not a matter of optimizing the assembly line. I think about that challenge, actually, because I'm not very good at it. My preference is to just have people in the lab who are already trained, and to give them a project that helps me, helps them, helps me.
For sure.
But things can get very complicated with large structures where everyone is doing separate things.
Sure.
And unless the leader is brilliant, so that they're on top of everything that's going on, and that can be very difficult. It just becomes interesting. I don't think that any part of our training really equips us to be in a large lab. How, so the way to organize that lab, the success I've had is that one of my earliest postdocs was a real people person. He would, very, very bright. He's very successful now, not unrelated.
Awesome.
But if you were in the lab with him, he would always say, I can show you how to do this. Come and ask me. We can do this together. So he was very good for the other postdocs in the lab. He was good for the students. And he had a calming influence, not very excitable.
So measured
and so I think that the key to having a good structure. And so I can't be in the lab every day.
For sure.
And because of that, I don't know what the day-to-day issues are, and sometimes people don't feel comfortable coming to me.
Sure.
But to have someone who can actually be there in the same space all the time, who's kind of at an intermediate level of authority and knowledge, I think is very useful.
Okay.
So if you want a bigger group, you have to have underlings who have the right sort of personality traits and are geared to being helpful. The lowest person we've had in the lab has been a high school student. The local high school, she went to the Illinois Math and Science Academy.
Oh, awesome.
I don't know if you know, but it's a residential school.
Yeah.
And they come and they do projects. They come once a week during their junior and senior years to do projects to get in the Intel or Simon's, whatever the competition is now. So I think that when you have someone that young in the lab, I actually wanted to spend some time with that person. So how does that person exhibit good traits? That person wants to come in with a good attitude, to learn, to do what they're told, not to have a kind of a resistance to learning. Some people have a tendency to fight, and particularly if you're very early on, you just want to listen. You want to do it. Of course, there's an importance to questioning. There's always an importance to questioning. But very often, I think, the lowest person in the lab has to come in with a very open attitude. And the reciprocity there is that they also need to have access to the top person.
So what I'm getting from that is, for both my questions, as a PI, if you want to exhibit certain leadership traits, you should be able to sort of be comfortable enough with your underlings to delegate, right?
Right.
So that
and you have to have good. And you have to have good. Delegation is important because you're really not there.
Yes.
As a PI, as a CEO or whatever, you're gone most of the time.
So that
have someone.
So that confidence in your students, to be able to give them some kind of autonomy to sort of find their way, but at the same time know there's some certain guidelines that you do prefer in your lab.
Right, and as you go down, to people who are newer in the lab, their obligations become more just to keep an open mind, to do what they're told. You can question on the sidelines, but very often the things that you're being told to do or trained to do are things that have been done in the lab hundreds of times. And
so they're tried and tested techniques.
Yeah, I have, we do the dissection of the ground squirrel retina.
Yes.
And I just tell people, you may think of your own way to do it, but believe me,
Yes.
I've done it 1,000 times. And when I say, move your arm like this, move your arm like that. When I say, move your arm like this. But
certain fundamentals you're
certain
more
fundamentals,
strict on.
I just,
Yes.
But on the other hand, I've had people who have learned the procedure and adapted it. They couldn't do portions of it. And at the end of the day, they were able to teach me something. So, it's kind of a balance. You want to train people. You don't want them to resist. But on the other hand, everyone can contribute something. Sometimes it's surprising. And you don't want to squelch that.
Innovation.
Innovation.
Yeah.
But on the other hand, if you have a tried and true method, you want them to learn
You want them to learn
first.
Okay. So at what point, that's actually interesting. So at what point do you think your student is actually skilled enough to be able to sort of experiment with new techniques, new sort of ways of looking at certain problems that, from time they've actually had certain fundamentals applied to them?
So I think that if they're able to do the standard technique, and therefore can proceed with their experiments with kind of standard material, they can innovate. They don't have to tell me what they're doing. But if they complain to me and they say, this isn't working, or I can't get it to work, I'm not going to be very happy with that. So, but beyond, so I want to give them a platform, and I want them to understand that that's what we're doing. But if they want to then go out and innovate, they can do that. Just don't tell me. Don't tell me until you
until you figure it out.
If you find something better, come and tell me. That's what I want to know.
All right. So how do you tie that back to when you were actually someone's student?
So, this is very interesting. These are all very interesting questions because the two labs I was in were completely sink or swim labs. There was very little.
Was that a sign of the times, or is it just?
I think it's so. It's the field, physiology. Is partially like that because it's usually one person recording, and you don't have lots of people who come together to do it. But it's also the pedigree of people. So my first and my second advisors, the graduate student and the postdoctoral advisor, he was an Italian gentleman who fled the Nazis during World War II or Mussolini, I'm not sure which, but he had to row himself down Italy. To the point where he could join the Allies and fight his way back up. So we're talking kind of a different generation of people. People who are highly self-reliant, who are probably not going to take the time to baby anyone. They're probably going to say, I did it. Now you do it. The other person in the lineage won the Nobel Prize, Alan Hodgkin, for identifying Hodgkin and Huxley, identifying the channels in their behavior that made up the sodium action potential. And they also were part of the war effort in Britain. Alan Hodgkin took time off to help design radar. And at that time, people just made their own equipment. You had to be really good
to be a scientist.
Now, you can kind of slide by. You can be a bio major. You can move up. If you're good at reading and maybe thinking, and you've got some talented people. Back then, I think they really had to know what they were doing. And so my advisor, who is the postdoc advisor, is a gentleman trained with Alan Hodgkin, and that's a picture from Stanford.
All right. And that's you, right?
And that's me. That's young Steve.
Haven't changed a bit.
Thanks. So Dennis went to train with Alan at Cambridge.
Oh, wow.
And the story's Dennis just wrote a biography in the Journal of General Physiology. If you want to read, he's probably 83 or 84 now, but he writes beautifully, and when you're reading this history of his time at Cambridge and interactions with Alan Hodgkin, the writing, it's almost like your own brain talking to yourself. So that's leadership.
Okay.
But the interesting thing was that he got to Cambridge, and Alan Hodgkin gave him a box of resistors and capacitors and said, build your setup.
Okay.
And it was to record from photoreceptors. And Dennis built it. And he started recording from the retina. You had to stick the electrode in the retina all the way through down to the photoreceptors, sharp electrodes in those days. He just didn't get anything for a year. Hodgkin was some sort of head of the Royal Society, potentially, and was out of the lab completely. And at the end of the year, Dennis went into his office and said, Sir, Alan, I would like to leave your lab.
Wow.
And the story is that Hodgkin said, shut up, and said, why? Why would you want to do that? And Dennis said, well, my project isn't going anywhere. And Hodgkin said, well, I'm going to sit with you every day, and we're going to make it happen. And he did. From that point on, he worked with Dennis. He would be solving equations in the back of the room while Dennis was doing his recording. But what I'm saying is that there's a tradition. I came out of a really sink or swim tradition. And neither advisor in their own, you kind of hope your advisor will help you with your career when you finish. And I think their philosophies were that they just don't do politics. That our help for you with your career is what we teach you in the lab. And we're not, once you get out in the world, we're not going to pull strings for you. So it's kind of very tough. And so I've changed. I've changed, to get around to your question, I've changed my point of view. And I do feel that, sink or swim is very tough. And you have to help people a little bit more. And then when they get out, you have to help if it's been good. And if you believe in them, you have to help them.
All right. So that actually brings something to my mind. Do you think at this point it's more of finding that perfect balance of sink or swim, but at the same time being available to sort of walk them through certain situations, right? Is that the modern day leader?
So I think, so you didn't meet her. My daughter was in lab. She's an undergraduate, just like you. Well, technically just like you. And she came into lab over the summer. She's a math major, but she does some of the animation. I think maybe Youngling, Chenyu. And what I tell her is that just don't ask the PI questions. The relationship is, we're going to give you your assignment. You go in and you do your best to figure out. This was Morris, one of the image processing programs. We'll tell you what you need to do. Do your best to figure out how to use the program. Do your best to understand the task. Then if you reach a roadblock, you come and you ask the PI what to do. But don't waste the bandwidth. Don't, if it's something you can do yourself with difficulty, please try it. And I think that's how the philosophy, and I told her, you're going to be very valuable to people if they can just give you the project. And you can say, I got it. I'll see you in a month. Got it. And then they will respect you a lot. That's the ideal. You're probably not going to meet the ideal. She didn't meet the ideal. She doesn't know neuroscience. She can't really think about, visualize it. But, it's very good. She went off back to her classes. She had a laboratory class at Woods Hole in Cape Cod. And she said, oh, my fellow undergrads, they're all like whining, to the PI, can you do this, can you help me, and stuff like that. And she wasn't doing that. She felt very proud. And,
I think that's actually a way to foster leadership qualities as well, right? So certain situations whereby the task might be difficult, but it's up to you to sort of, not be intellectually lazy about finding the answers.
That's right. And you can't make progress. I mean, the idea of having a person in the lab, their training is important. That's part of their training. But they can't be a force multiplier if you're always telling them what to do.
Sure.
Because then you kind of thought of it.
Yeah.
You spent the time to think the steps,
Yes.
and they haven't saved you all that much effort.
Okay.
You really want them to kind of save you effort so you can focus on the big picture.
Sure.
And that's not all of training, but I think that's where it's mutually helpful to the student too. They expand their faith in themselves to solve problems.
Okay.
Become more independent. Yeah, I like that.
Okay.
I like that.
All right, let's see. So wrap it up, actually, what's coming into my mind now is what you were talking about with Sir Hodgkins, right? So his student came up to him and said, okay, I want to leave your lab, right?
Yeah.
So I'd say at a certain point, a leader needs to know when to sort of jettison his or her student, right? So in that situation, the job is not done yet. The student is not ready yet, right?
Yeah.
So it can go two ways, right? The student can be ready and actually should be let go, or is actually...
Or they failed miserably.
They failed miserably.
And it's time for them to go.
Okay, that's interesting, because I was actually thinking of it in a different way. So you're saying there actually are situations whereby
there's
it's
students
not
who
gelling
just...
and it has the relationship has to end.
It's just not working.
Okay.
And so, I mean, there's one greater point I want to make, and that's that while I'm stuck in a web of responsibility and obligations.
Yes.
And, it sounds like nobody helped me. I suffered. I didn't sink. I made it.
Yes.
But in fact, there were people at key points in my career who helped me. I'm here for reasons, and I happen to know many of those reasons. And there's kind of an element of payback, and I should say that a good leader recognizes that they were mentored and other people helped them in their career. They might feel they're super excellent, but even then nobody lets you in. Someone, just because you're excellent, you can't fight your way in. You have to have allies. You have to have people who believe in you. And so I'm kind of in a web of responsibility. When I have people in the lab, those people, they've entrusted their lives to me in a way. And they can be kind of shattered for life if you spend a long time in the lab and you don't get a job as a postdoc. So there are a lot of things I can't do. I'm not Superman. But I have obligations to try and do the best I can to help them in their career. And I also have responsibility to the field. And that's an important part of leadership. You have to go out and you have to be on committees. You have to read grants and evaluate grants. You can't just take care of yourself. At a certain point, if you're going to rise up in the field, you have to be known as someone who gives back.
At least their time, right?
Their time and their intelligence, your wisdom, so to speak. And that involves, you have to demonstrate fairness and judgment. In our field, you have to do that in a semi-public way in the study section where the grants are read.
Wow.
Right? So I think that's important. And the other thing I don't like is that I don't like transactional interactions. I don't think it should. I don't think it should be. Well, you did something good for me. Now I'm going to go out and do something good for you. I think that people can make a good effort and fail, and they can still be impressive people. If they fail, it's probably my fault. So, one of the most crushing, when I interviewed for I was in an MD PhD program.
Okay.
So I interviewed, I forget what university, Wash U, very good program.
Sure.
The professor, also an MD, PhD, I interviewed with,
Mm-hmm.
Said, well, I like you, but I look at you and I wonder, what can you do for me?
Ooh.
If you're in my lab, what exactly would you do for me? And I see that actually you can't really do anything for me. So, unfortunately, basically the bottom line is, unfortunately, I'm not going to, write you a good recommendation to be in the program. And that just stuck with me as, just kind of very nasty. This should not be, you're not here, I don't think Youngling, I think she feels you're capable of furthering the purpose of the lab.
Yeah.
But, I don't think it's that transactional in that way. And so I don't think that a good leader really deals in transactions.
So on your part, was that fuel, or was it something not to aspire to do or to be?
I have a lot of negative. I don't like to teach by sink or swim, although I do to a certain extent.
Yeah.
And it's really, we're kind of in this together. If you join a lab, you're kind of at our level, someone's not being a doctor, they're not being a businessman, they're kind of sacrificing a lot of their life, so you got to take it seriously. So it shouldn't be transactional. There's kind of a higher level commitment there.
Okay.
Not
So-
what they can do.
So this mission sort of transcends, the material. It transcends.
Yeah. You're dealing with people,
Yeah.
and you're dealing with potential,
Mm.
and you do have some obligation to provide for their potential. I think there are limits
Okay.
to what you can do, right?
Sure.
Because this is not the sainthood or anything. I'm not here to sacrifice my career, but I do want to provide a good environment so that people can move ahead with their careers, and you can all move ahead together. It's tough. There's a lot of conflict. I suppose there's a lot of conflict of interest involved here.
Yes. Yeah.
You really want to use the students and throw them away when you're done. Keep them as long as they'll stay.
Okay.
And then, throw them out so they're not your future competition.
All right. So that's interesting. So I think that actually
I'm
brings
kidding. Right?
Well, no, it's
But there are people who have that philosophy.
Yeah.
Right.
Yeah, but at what point do you actually do let the student go? At what point do you feel the student is ready, even though they're done with their program, but at the same time, at what point is it that they're actually their own person, and you're comfortable with them actually carrying your lineage?
So I've had mostly postdocs in the lab.
Okay.
But I think the same would go for a graduate student. Kind of the feeling is they have to do one paper where it's based on my own work, and I kind of know what the answer should be. It's something I've observed that I think is very interesting, but it's a real phenomenon. They don't have to figure out what they're going to do. I'll give them a project, and when they complete that, then they have to complete a project that is more based on their own intuitions. I would give them a second project, but it's kind of expected that they can go off now, and they can, whatever their own vision is, as long as it's within the scope of the lab, they can do it.
All right.
So that would be, the ideal case.
Okay.
There are some people who come here with different skill sets from me, who right off the bat are probably doing more independent work.
Okay.
But I like to see some independent work.
Okay.
Someone has to be in the lab minimum of 3 years, 3 to 4 years probably. It takes 1 year to train, and then 3 years to work a project out.
So what I see in that is it's kind of like a situation of an artisan, right? That has his touch in his work, right? He can see himself in his work. So is that what you're looking for? You want to be able to see yourself, or at least see your work, at some point in your students' projects?
So I want, yes and no.
Okay.
I mean, you're actually referring to the first situation.
Yes.
And initially I thought you were talking about the second situation.
But
I don't want to see my work in their project. I actually want to make sure this is the sink or swim aspect. I actually want to make sure that they have a project that will work. So they're coming here and they don't know much about the preparation. I don't want to stack the deck against them.
Yes.
You have to learn how this works, and you have to make up your project. If you learn how it works, I'll give you something, and it's going to be good for you.
Yes.
You'll get a publication.
Yeah.
And I can help you because I think I know what the answer is. And so you're not completely going out on a limb. And that's the first experience. So that's sort of the training.
Okay.
I kind of know what the answer should be. And if they're coming to me and it's just something wild, or if they're not seeing the phenomenon, I can correct it. But then at a certain point, I trust them to go out and do something where I'm really not sure what the answer would be. Okay. And they kind of have to rely, I'll talk to them, but I'm not in there while they're doing the experiment.
Okay.
I have to really rely on them to interpret what's going on correctly. And so that's the part where I consider the expression part. They're not necessarily expressing me, but they're expressing themselves. And if a person can't really express themselves, then they are not really succeeding in the lab. And failure to express yourself, what causes failure? Failure is caused by pessimism and skepticism. Actually, there's a certain amount of skepticism that's healthy, but I have people who, at a certain point, you have to look at the cup and it's half empty, but it's really half full. So you're doing the experiment, and you're wondering, no one's ever done the experiment before. Is it working? And certain people can look at the result and say, it's working. It's not there yet, but I see it. I see it. It's the answer. All I have to do is I have to change a few things, perform some controls. It's going to be good. Other people can look at the same piece of data and say, oh, this is not the expected answer. This project really sucks.
Is that something you cultivate from experience, or
no? These are just people. These are types.
Okay.
These are types of people, and the people who I've had to ask to leave are often people who just self-sabotage. The answers are probably there, and they're kind of lost in the noise. Just, it's 90% of what I think it should be. It's not 100%. There's something really screwed up here. I can't figure out that 10%. I'm done. And that's probably, it's the people who can sometimes an experiment doesn't work. You're wrong.
Sure.
And, you also have to be able to say, I'm wrong. And my recommendation is that someone should reach that point very quickly. Experiment. You don't want to spend a lot of time optimizing only to find out it's not that way.
It's not that way.
So my own feeling, I call it N equals 1.
N equals 1.
So when I do an experiment, I try it once. And if it works, even a little bit, I'm pretty sure it's going to work. And if it just doesn't work, if this pharmacological agent doesn't block, then that's it. If it was an otherwise fairly decent experiment, I'm just not going to do it anymore. So you have to, there's kind of two parts of it. You have to decide quickly whether something is going to work or not. And then, you have to get that positive feeling. This is good. I don't know where you sit on that spectrum.
Well, the way I see it, it kind of reminds me of Peter Thiel's sort of theory of 0 to 1, right?
Okay.
So the whole idea of you have nothing and you actually kind of transcend it to something. Because again, we don't have to go past one, right? Yeah. All the answers are actually between, naught and one, right? That's what I'm. That's what
It's, but there are just certain people by their character who just can't. They're throwing up their own obstacles. And I have notebooks that are just thick with data.
Yeah.
But ultimately, I had to ask them to leave the lab because everything was, I just can't do it. This experiment is not working.
Interesting.
I could tell you stories, but if I start, it's very bad. One guy, probably the smartest graduate student I had, an MD, PhD. Basically, if he hit a problem in his experiments, he would come into my office and say, "Give me 5 things I need to do." And then he would do them, and he would come back. He would say, "None of them worked. What should I do?" I don't know. Find some way to make it work.
Yes.
And for that person, the idea that you could ask the authorities and the authorities wouldn't know the answer was very discouraging. That person. This is science. Yeah. Sometimes there are a lot of things it takes months or years to figure out.
That's right.
And there's no book. That person went into computer programming because you can always solve the problem.
You can always solve the problem.
There's always some solution somewhere. He works for software. He got his PhD, he dropped out of medical school, didn't get his MD, and went to work with a software company that makes the software package. Because he was on the phone with them every day trying to do programming.
That's interesting, because I was talking to Andrew about that because of my situation, and he's telling me that there's actually a lot of computer science students that are transferring into neuroscience nowadays. Is that
so? There's a lot. We were just at neuroscience. There's a lot of machine learning algorithms going on, and a lot of that stuff you can solve in one way or another, right?
Yes.
There's always more data or whatever, tweaking the algorithm, and that's computer programming. Actually doing wet science is a whole different feel to it. You could get very frustrated doing wet science.
Oh, yes.
and not know what the hell is going on. And not everyone is really cut out for that. Some people really like it. Today's challenge. I'm going to go at it again today. But other people are like, I don't know what I'm doing. I don't know how I solve this problem.
How to solve it.
So that's the thing. So you
2 machine learning classes next quarter at McCormick, because I actually do want to go that direction, machine learning and artificial intelligence.
Okay. Yeah. So they had this woman. Yang Ling said, he's always talking about Fei Li.
Yes,
Fei Li. And so Fei Li, I didn't know that.
Okay.
But I attended her talk.
You did?
Yes. She spoke at SFN. And her lecture, you've probably seen her lecture if you
all
like
of them
her.
Probably.
Yes. So I don't think this is any different from that.
Yeah.
Someone had asked her at the end of the lecture,
do
these, when you finish, when the machine learns, does it end up in some way like the human brain? Is the wiring or the structure or the algorithm how we would do it? I forget what the answer was. I don't think she could answer that question.
Well, I'd say no, because at that point we probably would have a general human intelligence, which I'd say we probably won't in my lifetime. I don't know, But so the thing is, with these weights in these machine learning models, right?
Yeah.
So the first time the information goes through the model, it actually makes arbitrary values for our weights, right?
Okay.
So then as it learns, right, it actually creates some sort of representation of the information, right?
Okay.
You know, so because you have a machine learning model that works on maybe recognizing, I don't know, certain retina neurons, right? Okay. Can we use that same model to recognize human faces?
Yeah.
You know, I don't know. Like, the accuracy isn't
actually going to be,
you know, that high, right?
Yeah.
So, yeah, we're far away from general human intelligence. But, I mean, is it an algorithm problem? I don't know. I mean, these are sort of, you know, PhD level questions, right? Is this an algorithm problem? Are these hardware problems? I don't know. But, you know, it's a very interesting field, and, you know, like, I mean, there's just so much going on in it. And, I'm super interested in what Fei Li is doing. Yeah. She focuses more on computer vision.
Okay.
Right? Yeah, so, which, I mean, it kind of relates to what we're doing here.
Yeah, so she has a human-centered institute.
Yes,
for
seems
Stanford. kind of naive.
Yeah.
Human-centered
Institute for AI. Intelligence. Human-centered intelligence.
Seems a little naive, but I guess you have to be.
Yeah.
You have to attain that because you're in a scary field in a way.
Yeah. And what I do like about what she's doing is the fact that she's trying to democratize AI. You know, she's trying to get more diversity into the field, right? Because it's still human beings that are training these models, and as flawed human beings, we're actually going to insert our biases into these models, and you know, that is going to be a very scary world, right? Automated discrimination.
Yes,
you know, so.
And even mental discrimination. We like people who think like this.
Yes.
You know, think like programmers.
Yes.
You know, we'll pass them through.
We'll pass it on to our models, and that might be a big problem. So what she's trying to do with, you know, her efforts are commendable. Now, is it something she can do? I hope so.
Okay.
But she's an I think she's actually on a sabbatical right now at Google.
Is that so?
Yes,
okay.
Yes.
Just up the road.
Yeah, not too far.
There's a vision researcher at Stanford, Brian Wandel. He's in the National Academy, and he does this for industry. Basically, he's a visual psychophysicist. He's developed a program for simulating all and incorporating all the knowledge that we've gained over 400 years about the nature of light, and the visual apparatus, and then absorption in the retina. And so he begins with I don't remember the name of the ray tracing program, but it's kind of an authentic ray tracing program where surfaces have the same properties and reflectance that real surfaces have. And it's not the program that a game corporation company would use.
Okay.
They cheat all the time.
Okay.
And so he begins.
So this is not aesthetic. This is more.
He creates a realistic 3D world, and then his program will ask the photoreceptors, "What is your photon catch per unit area at this point?"
At this point
in the retina, and so he's able to compute photon catches, and for him, he fits in well with industry because people are concerned with virtual reality.
Yes,
they want to know exactly how to create, psychophysically, how to create an image that people perceive as real, with this kind of goggles and optics and all this stuff.
Interesting.
How do they judge distance? So Brian Wandel is there, and I'm sure he and Fei must
chat
about that. It was an interesting poster in neuroscience. It's a former colleague of ours. He moved to University of Maryland. I'm mentioning this because, maybe it's the opportunities. Maybe you've already gotten some ideas about machine learning from the work over there.
Yes.
But this is something that Yang-Ling and I pretty much decided to do. This guy went about it a different way. His interest is in labeling an individual amacrine cell, fixed tissue, and then staining the synapses.
Okay.
and counting the synapses of just a single neuron using a super resolution technique, so you can really resolve individual synapses. It involves serial sections and reconstruction of the cell. But then his student, who must be an engineer, created a program for recognizing synapses, because each neuron you do this on might have 20,000 synapses. You don't want to sit there with your clicker counting through by hand. So the synapses, the neuron, surface of the neuron might be blue because it's labeled with channelrhodopsin and there's an antibody to channelrhodopsin. And then pre- and post-synaptic faces are different colors, red and green. And the whole inner retina, the cell you haven't labeled, has all these pre- and post-synaptic clusters. But the cell you've labeled has them too. And somehow you have to associate them with your cell and then count them. And some of the judgments can be very difficult to do. I don't think she took a machine learning approach. I think she just kind of programmed, if you're located here and there relative to the membrane, then we'll accept you as a synapse. So she went through
a more symbolic AI sort of situation. So if this, then that. So probably a series of if else sort of.
I don't even know if it's AI, but she would look at each. If you are a red structure, are you nearby a green structure? If you're a red and a green structure, are you nearby a blue structure? And so kind of
that's interesting.
Discrete programming. Not training. There was no real training in there. But we're interested in doing something like that. We're looking. In our case, it's not as dramatic, but we have this rabies virus that labels the pre and goes to that labels the postsynaptic cell and goes back to the pre. Everything is the same color, but we're probably going to use super resolution and stain for the synapses to try and find that point between the two neurons that the synapse. So you can see where one cell begins and the other ends.
That's interesting.
And so it's sort of a similar kind of training, a little bit more constrained because we're only looking at some spots. But you can imagine expanding it to all of the spots associated with the neuron.
That's very interesting. Is that something I can talk to?
That's something you can mention to Youngling, that you can build a synapse recognition program.
Because all I need is just data sets. All I need is actually just images, and I label them, and I can actually train a model to do that.
And I don't think anyone has done that. People may do it in the visual cortex. I can't say no one has ever done it ever. But in this particular instance, it wasn't the way they went. And her methodology is probably a little flawed for technical reasons. And so it might be better to have something where you didn't have to program a discrete algorithm. For instance, it turns out that at the synaptic contact, the cell you're interested in is not labeled. The label is excluded. The channelrhodopsin is excluded from that region. So it's a little difficult to actually say whether your presynaptic proteins are really associated with your cell or not, because they're actually sitting in a blank space. And so she has to, in her program, it's not so much overlap, but are you one pixel or one group of pixels lateral? And I think the program probably has a pretty good error rate. I'm guessing.
That's
so that would kind of that would be kind of a nice intermediate level thing. Without, you could probably do signaling too, but signaling might be more difficult for training.
Signaling would be
some of Andrew's signaling would be the calcium, the GCAM.
GCAM.
Like under a given stimulus. Could you, machine learning would be, could you read out what the stimulus was from the calcium signal? but that's maybe gets a little bit more toward information theory or something. You know, what is the noise, the reproducibility?
Okay.
I don't know if there might be ways of analyzing signals that involve machine learning.
Yeah.
But then I don't know if that would be. Well, this is my philosophy.
Sure.
I don't know if that would be the first thing someone would ask you to do, because that project could fail or end up, garbage in, garbage out, in a way.
Yeah.
But
that's interesting. I'll talk to Young Ling about that.
Yeah, the synapse recognition, I think, could be very useful for her. She's all these Cree lines, and they label individual cell types and being able to kind of notate which synapses are associated with them. Yeah,
because I know Andrew's soon going to start doing experiments with the rabies virus.
Yeah, so they've asked you to get vaccinated.
Yeah.
You could do it. You could do it. It's generally useful. You'd be amazed. You can go caving. You can explore caves now without being afraid of getting bitten by bat.
Yeah. So my insurance actually only covers like $800 of it. So it's
should pay $0. So that's the key. No one in your situation ever pays their own money. So Youngling will have to do it from her grant.
All right.
She'll have to find a way to talk to the administration.
Okay.
Because you're doing that for the lab. I was only joking. It's not for your own health. Here, doggy. I don't care. So you don't have to worry about rabies.
All right. Awesome.
Yeah. So that
interesting. Yeah, because I'll definitely talk to her about that.
Yeah, and Youngling, she has her moods. Maybe she'll take your head off and say, "No, you're not going to do that." But see how it goes.
For sure. So
did I give you enough?
Oh, more than enough.
Sorry about that.
No, it's awesome.